Hello students, today in this fourth lecture of Applied Environmental Microbiology course, we will be continuing where we left off last time on cell structure and then proceed ahead to talk about the informational molecules in the inside the cell. So last time we talked about cytoplasmic membrane and I told you about the bilayer in bacteria and monolayer in archaea and how the external and the internal portions are hydrophilic, they love the water, and the inside portion is hydrophobic. These are fatty acids, long chain of fatty acids. Now today we will study about cytoplasmic membranes, how they differ in bacteria and archaea, little bit more in detail. So let's take a look at this beautiful picture here. This picture is a cartoon showing a cellular membrane of a eukaryotic cell, and many of the elements in this cellular membrane are shared by bacterial cells also. So many of the features in this are similar to what we see in bacteria. So let's start with this blue protein here. Now notice that this protein transverses the inner face of the cellular membrane with the outer face, and this such kind of membrane proteins are called as integral protein or intrinsic protein. There's another integral protein here in purple color. Many times these integral proteins are used for transporting compounds in and out of the cell. So many efflux pumps, for example, are integral protein, and this one particularly is shaped to look like an efflux pump. Now we can also notice that there are sugar side chains on the outer face, and these sugar side chains, they have very specific conformation, very specific features, both chemically, physically, and in stereoically. Now these sugar chains, they act as sensors. So now the cell using these sugar chains can sense different kinds of particles and compounds in its environment. It can find out where the glucose concentration is increasing, what kind of virus and bacteriophage are present in the environment, what kind of compounds. Microbes can also talk to each other, and they can sense pH and change in other chemicals. So these are very, very important, the sugar side chain. One of the recent applications of sugar side chain is that we are targeting them because they are very specific to each cell. Let's say bacterial cell, Staphylococcus aureus cell will have a very specific sugar side chain on it. It can be used as a marker, as a target to design biosensors. And then you see here purple balls. These purple balls are the hydrophilic part of the cellular membrane, and they are facing the aqueous environment. And then here we have blue lines, which are hydrophobic portion, and the blue lines, they attach to each other, they stick to each other, and they make bilipid membrane. You can also note here that look here, there's another peripheral protein. So not all proteins have to be integral proteins. Some of them only face either the exterior or the interior part of the cell. So exterior could be as a sensor or as to respond to different kinds of bacteriophage attacks, and the internal are used for different purposes like receiving certain compounds or just sending the message or other things. Now notice here in this yellow dots, this is specific to eukaryotic cells. These are cholesterol, and we bacteria have equivalents of cholesterol but not cholesterol itself, and this helps in giving certain amount of fluidity to cellular membrane because cells need to move. Locomotion is important for different reasons. Very few cells, very few rare eukaryotic cells have complete rigidity and do not have much of cholesterol or cholesterol-like substances to give them flexibility. Now here we have a lipid bilayer example, and this is shared both by bacteria and eukaryotes, and whereas in archaea we have lipid monolayer. I also talked in the previous class about gram-positive and gram-negative cell. Briefly, gram-positive and gram-negative cells are classified on basis of how they respond to gram staining. So Dr. Gram, he discovered that certain bacteria were he was able to stain them using gram stain. He called them gram-positive cell, and the gram-negative cells he could not stain. The reason is that the stain actually interacts with cytoplasmic membrane and is visible then under light microscope or bright field microscope. So in gram-negative cells they have an additional layer of periplasmic proteins which do not allow staining, and thus they are called gram-negative cell. Even though gram-positive and gram-negative cell classification is a classification based on the cellular membrane and structure and not genetic structure that people often talk about nowadays in microbiology, it is a very important and informative classification. I also shared information about lipid bilayer and lipid monolayer, and how lipid monolayer of some archaea give them particular strength and resistance to heat and extreme environment. This should remind you of one of the previous lectures where we shared information on how archaea can live in extreme environments such as extreme high pH or low pH, high temperature and high pressure. Here's another photograph of cytoplasmic membrane that I showed last time. What is very important for you to notice this time is the size of the cytoplasmic membrane. It's at the range of 6 to 8 nanometer, which is very, very small. And thankfully we have techniques such as TEM and electron microscopies that can give us beautiful images of cellular membrane. Enough beautiful that now we have such beautiful detailed information and we can create such nice graphics. And notice here on the top left of the slide that the hydrophilic end of the cytoplasmic membrane in bacteria is often glycerol with a nice phosphate end. In archaeal membrane there is a difference. The archaeal membrane and bacterial and eukaryotic membrane differ from each other in a very essential manner. In bacteria and eukaryotic cytoplasmic membrane, the fatty acids, so long chain fatty acids, are linked using ester bonds. So ester bond is when we have C, a ketonic bond here, double O, one O, oxygen here. So this is an ester bond. Fatty acid attached with ester bond to the glycerol phosphate group. In archaea, they are almost always attached by ether bond, so they are missing this double bond O here. The other important difference is that, as mentioned earlier, archaeal membranes are monolayered and bacteria and eukaryotic membranes are bilayer. Let me just spend a minute here to explain, to remind you of what eukarya is. So earlier we talked about 3 domains of life. So let's take a look at what eukaryotic cells are. We believe that life started from a particular kind of cellular structure, and we can refer to it as last common ancestor. So this is an ancestor that's common to everybody, and we don't exactly know who came first in terms of prokaryotes and eukaryotes, but we believe it's definitely prokaryotes. But among archaea and bacteria we do not know, but let's say archaea were the first to arrive. So we have archaea. So certain amount of last common ancestors specialize themselves into archaeal domain. Some of them split into bacteria that we know a lot about. The archaea are nearly a recent in discovery, and then after billions of years they split into higher forms of life called eukarya or eukaryotic cells. So this is a revision of what we talked about in the first lecture, but I think it's an essential revision. So bacteria and archaea are often coupled together as prokaryotes, to contrast it with eukaryotes, which means true nuclear cells. The difference between eukaryotes and prokaryotes is that eukaryotes have nuclear membrane and cell organelles. So if this is a cell, the nuclear material is stored inside a nucleus, and then they have other organelles. So as I mentioned in the first lecture, that these are cells that have multi compartments inside them. So it's like a building with many rooms, whereas prokaryotes do not have that luxury. It's a single cell, single room house where everything just floats around and interacts with each other. They're usually much smaller than eukaryotes. Now interestingly, eukaryotes and bacteria have some commonness with each other. For example, their cytoplasmic membranes are very similar. Both are attached to their hydrophilic end using ester bonds, whereas archaea uses ether bonds. Another common thing between eukaryotes and bacteria is, again coming back to their cellular structure, is that in eukaryotes it's very unlikely to find anything else except fatty chains, simple fatty chains. In archaea, however, we can have fatty chains that have other appendages attached to them. In fact, sometimes they have cyclic compounds that are attached to their fatty chains. An important difference now between eukaryotic cell and bacteria and archaea is, as I mentioned earlier, they have nucleus and other organelles. Bacteria and archaea don't. Now To throw some more relevance and light about it, most pathogens that afflict humankind, or at least the ones that we know about and are concerned about, are bacterial. Archaea are nowadays found very often in extreme environments such as deep under the earth, in hot sulfurous springs, in deep ocean volcanoes. Eukaryotes is almost every kind of life we see. So this would range from algae, fungus, chimpanzee, trees, tiger, ourselves, insects, butterflies, all of them will come under eukaryotes. So just imagine this way, that the diversity in eukaryotes is considered to be minuscule, very small compared to the diversity that we know in bacteria. And we also have reasons to believe that we have only scratched the surface of bacterial communities. So there is unprecedented bacterial diversity that we have not seen yet. We know this by genetic sequencing and fingerprinting of bacterial communities. So imagine if the eukaryotic diversity that we see, we talk about diversity of ecological life forms, man, we have these many species of trees and plants and insects and animals, and even within humans we have such diversity in terms of how we look like and how our bodies are structured. We cannot even begin to imagine that if we had eyes to actually see bacteria, how much diversity there would be. And obviously still much is not known about archaea. Now coming back here, we notice that despite differences in their in the membranes of archaea, eukaryotes and bacteria, they still have very similar structure, in sense that the inner side of their cytoplasmic membrane is hydrophobic and the outer ends are hydrophilic. They love. So here hydrophobic, outside hydrophilic. Similarly here hydrophobic and outside hydrophilic. And this is important to survive in the environment, to keep the insides of the cells intact and together, and also give it selective permeability. So not everything that's inside can go outside the cell, not everything that's outside that can come inside. They have to get the permission from the cytoplasmic membrane, which brings us to transport. So if we have a cellular membrane or any kind of membrane that we need to transport stuff across, in microbiological molecular scale, we have the first phenomenon, diffusion, which is normal transportation. But we can notice that when we have passive diffusion or just the Brownian diffusion, we do not get very high rate of transport. And when the cells actively transport or facilitated diffusion, they are actively transporting, they are spending energy and chemicals and other things to get things in or to throw things out, then the rate of transportation increases. So this is inertia, this is action. So when it comes to diffusion, which I have bulleted as 0, this is just generic diffusion that's always happening. Except for water, other molecules find it really hard to diffuse inside and outside the cell. In events when cells need more water than what they will receive by diffusion, or at a higher rate than what they will receive by diffusion, they use aquaporins, and we'll talk about them. Now facilitated diffusion or active transport is generally classified into 3 groups. One is simple transportation, the other is group translocation, and then is ABC transportation. Now in simple transportation, let's take a look here. Simple transportation uses proton motive force. Now remember, I told you that the outer layer of cell membrane will have positively charged proton, inner layer will have negatively charged OH minus. So the cell makes use of this electron gradient, remember, like a battery. So it makes use of this electron potential to transport the substance here in red inside. So when it transports the substance in, the proton travels from here to the inside, or in other words, the electron travels from inside to outside. So the cellular potential is reduced by 1 charge and the substance has been transported inside. This is simple transport. Now in group translocation, this is really neat. In group translocation, we have our substance waiting outside to come inside using this integral protein. By the way, this brown thing is integral protein, and here we have phosphoenol pyruvate waiting to phosphorylate the substance. So basically this substance is pulled in, it is phosphorylated, and it is chemically modified when it comes in, but it still comes in. So here the energy is chemical energy of phosphorylation. In ABC transportation, this is usually it's common in gram-negative cells. We have periplasmic proteins which bind with the substance that we want to bring in. So these, they bind with this protein bind with the substance, they bring it to the integral membrane that wants to transport it, and one ATP is utilized. Now remember, ATP is the currency of energy. So how in our human societies money is the currency with which we do our transactions, in biochemistry ATP, at least in biochemistry, ATP is the currency of energy. So ATP loses its one phosphate, and what we have is the substance coming in. So cell expends one of its ATP, converts into ADP, and the substance comes in. Now it's very important for you to note and think about these four different kinds of transportation across the cellular membrane, and wonder, in all these four, diffusion, simple transportation, group translocation, and ABC transportation, what is the source of energy of transportation? We know it by law of mechanics, any action requires some energy. If we are breaking through the inertia, thing is lying outside, we want to bring it in, we want to do work, any work requires energy. So what is the source of energy of diffusion? What's the source of energy of simple transportation? What's the energy for group translocation and for ABC transportation? The other thing I want you to understand and ask yourself is, for all these four kinds of transportation, in which of the cases is the substance that's transported inside not modified, and in which of the cases the substance when it comes inside has been modified. Now here's another beautiful picture with the text box here saying uniport, antiport, symport. This informs you basically about the direction of transportation. Not only do we bring things from outside to inside, but we can also take things that are inside outside. So uniport would be unidirectional outside inside motion. Antiport is one substance comes in, another substance is thrown out. Symporter is two different substances are brought in together in synergy with each other. And as I said before, for water transportation we can have passive diffusion, we always have passive diffusion, but we also have specialized integral proteins called aquaporin. These are special doors for water to come in and go out. And through the cytoplasmic membrane, if there is a chemical that is small and it is hydrophobic, it can diffuse, but if it is polar, if it is charged, it needs active transportation. Alrighty. Now this is where we will complete our inquiry on cell structure. This is a slide from your first lecture, and I hope you are reminded of it. So we have two pictures on the left panel. We have a prokaryotic cell and we have a eukaryotic cell. Now in a prokaryotic cell, you can see there are no cellular compartments apart from cytoplasmic membrane. In eukaryotic, we have all these different structures, and I want to briefly go through them and tell you what they are. So here we have nucleus in prokaryote. This yellow liquid is cytoplasm, which is an aqueous mixture of biochemicals that are necessary for survival. So it has carbohydrate, it has proteins, it has some polar compounds as well. This green 8 here is the plasmid, and we'll talk about it in the next lecture in more detail, but for now it's important to know that most of the genetic material, genetic material, what is genetic material? Genetic material is the material that stores information about the cell. It tells the cell how are you going to live, what are you going to do. So most of this genetic informational material is stored together for prokaryotes in one long circular chain. However, it can have some extra material that is stored separately, and one of them is plasmid. In eukaryotic cells, we have a more complex and a multi-layer structure. So I have mentioned this before. The yellow bodies here are called Golgi complexes, and it's good for you to know that they're used for processing, even preparing, transportation, and separation of different kinds of proteins. They have many purposes, but protein transportation, sorting, and processing it are the main functions. The green things here, endoplasmic reticulum, sometimes they join the outer end to the nucleus, and they are used for many reasons again, such as protein formation and sharing. The pink structure here is a mitochondria, and this is something I want to highlight. In this particular cell we have 1, 2, 3, 4, 5 mitochondria. Now mitochondria is interesting because it has its own genetic material. It seems to have a mind of its own. Its genes are different from the genes inside nucleus. Its genetic material looks and behaves differently than the genetic material here. And what would be very interesting for you to find out is that mitochondria, by the way, mitochondria is the powerhouse of the cell. it gives it its currency. So it's like a bank that prints notes for the cell. So mitochondria is slightly bigger than prokaryote. Mitochondrial genome or genetic material or the informational material resembles prokaryotic material, which gives an idea to many microbiologists that mitochondria was once an independent bacteria which was engulfed by an eukaryote, and now it's an essential part of all eukaryotic cells. And this is me giving an idea of difference between mitochondrial size, few micrometers, eukaryotic cell size really big, and bacteria up to few micrometers. So many of the organelles inside eukaryotic cell are actually size of prokaryotes. And then today we are just gonna talk a little bit about arrangement of DNA in microbial cell, and in next lecture I'll give you more detailed information of what DNA is, how it works. For now it's very important for you to understand that there are two kinds of material in cell. When we talk about cellular structure and the components, biochemical components of the cell. One kind are informational and the other kind are non-informational. So all carbohydrates, the cytoplasmic membrane, and many things in the cytoplasm are non-informational because they don't inform the cell how to go about life. They act like sensors that respond to information. They don't have information in themselves. But the cells have a highly sophisticated DNA or genetic material in them that inform the cell what to do, when to do, how to respond. So it's basically like the code of life. It's akin to library of the cell, and it's only, but here's the important thing, it's a library of cell. It tells the cell the information that it needs, but only the information that it needs, and the information that it does not need just sits quietly within the genetic material. So not all the information inside the genetic material is provided to the cell at any given time, but when it is appropriate, only then it gives the information. Now how it does that, how it interacts with the cell, how it responds to the environment. Let's say environment is glucose rich, it has a lot of glucose around it, so the bacteria can process glucose. So bacteria would like to have the information, or genetic material would like to tell the bacteria, hey, there's a lot of glucose, use it. Now how would genetic material know that there's a lot of glucose? The sensors outside the cell would send chemical signals informing or triggering the genetic material to convert into proteins which will allow it to process the glucose. Same thing happens in eukaryotic cell, only that their arrangement of genetic material is very different. Like Earth has a core which holds inner core and outer core which holds liquid lava. Similarly, eukaryote has an inner core nucleus that holds its genetic material, and they have a very sophisticated system for transporting information into the cell and interacting and making sure that the cell survives. So today we will only talk about basically what genes are, what genetic elements are, and what genome is, and in the next lecture we'll go more in detail. So for now what you need to understand that gene is the smallest size of information that a cell can decode. So this is like a sentence. When it comes like for language, what a sentence is, it's the minimum complete set of information. So I have given names of some genes here, and we'll talk more about them in detail later. But MCRA, DSRA, 16S-rRNA, VAN-A, these are names of genes that serve a particular information to the cell. So MCRA, this particular gene would tell the cell to make a particular enzyme that will allow the cell to produce methane and create energy. Similarly, DSRA would tell the cell some information that will allow it to reduce sulfate and get energy. Now many of these genes often almost always are clustered together and joined together to form one big genetic element, and the most popular among them are relevant among them are chromosome and plasmid. And here is one of the early pictures of chromosome of E. coli, and together all the genetic elements that are present in a cell are referred to as genome. And in the next lecture I'll go in depth about gene and genetic elements and genome and how they inform the cell and how this beautiful molecular biology of cell works and what are the promises of this molecular biology. Until then, thank you very much.